Namaste to all. Welcome to the next session on drug designing. So in this session, we are going to see about drug targets. This is a very important topic. Why? Because in this particular topic, we are going to discuss about the receptors and how this helps for the target selection. In pharmacology, if you see, target plays a very vital role because whatever the different molecules are there, which you are going to keep it against any receptor or on the particular bacteria or any other cell surface, they will be having a receptor like a cup-like structure. So what happens is if a molecule is a round-like structure, it has to fit properly so that what happens is there is a complex which is being formed by which it can inhibit the reaction or it can initiate the reaction. Both the things can happen. So this actually helps us to understand that how the process is actually taking place here. So in this process of drug target, the first and the foremost thing what we have to understand is in the drug discovery, it begins in the laboratory with the scientists of various functional areas working together to identify cellular and genetic factors that play a role in specific disease. So in every laboratory, wherever the research is happening, they actually concentrate on this aspect only, that how the particular molecule can work and can actually help us to identify the particular molecule which can bind with a particular receptor and can stop the particular function, thereby preventing us from the ailments or diseases. So the identification of the target and sources, there are 4 steps are there. So we will see this in detail. So in this particular topic, the target selection, we have to understand that the target selection in drug discovery is defined as the decision to focus on finding an antigen with a particular biological action which is anticipated to have therapeutic utility. See, when we are formulating this particular molecule, we have to understand that it will be designed in such a manner that the receptor which is present that has to bind at this particular target area. So this receptor acts as our target. So the target identification plays a very vital role. Here, to identify the molecular targets that are involved in the disease progression, how the disease takes place with this particular target area and how it is going to take place, that we have to understand. And target validation. Validation means to prove that manipulating the molecular target can provide therapeutic benefit for patients. So what happens in the normal case or the scenario is, if a target is there or a receptor is there, if a molecule comes and binds, so what happens is there is a cascade of reaction which takes place inside. So either it can change the shape, like it can modify its shape and thereby starts the cascade of reaction, or it can actually bind and initiate a process by which the other molecule can get activated and can initiate the process of changing or conformational change, and thereby the process can be initiated. So to get the drug action, it is essential that sufficient concentration of the drug reaches the site of action. So that means, see, for example, the dosage level is very much important here. So we have to estimate the dosage level so that the sufficient quantity or the concentration can reach to the particular target site. Remains there for a sufficient duration because you know that the receptor is there. So for the example, the shape is like this and opposite side only it is like this. So what happened? It has to turn around and it has to fit, something like that. So what happens? It will take some duration so that it can interact properly and the tissue is susceptible to the drug action. So what happens when the complex is being formed? There will be an action by which what happens is the tissue which is present here, the cascade of reaction starts and the drug is working there. So it becomes susceptible. The tissue becomes susceptible. That means it can react to the particular drug. If it is not acting at all, then it is called as resistant. So here the main principle for any of the drug discovery is the tissue or the site should become susceptible to the particular molecule. So the magnitude of the drug action is proportional to the concentration of the drug at the site of action. So what happens here is how much the drug you are providing, whether it is very high drug, high dosage drug or concentration, how it is there and how it is going to react. Higher the concentration, you cannot say that it is going to work on the site itself. It can be a minute amount also which can work. So like nanoparticles, which can work on the particular site. So the nano size also plays an important role. Micro size also plays, or milli size also. So whatever the size is, it depends on the particular site where it has to go, or based on the tissues where it is going to work there. So the receptor mechanism is very important to understand the action and effect of a drug. So the receptor, as I said, it plays a very vital role for the action as well as the effect of the drug. So in the drug selection process, if you see the biochemical classes of the drug targets, that are divided into different aspects and percentages, like G coupled receptor, G protein coupled receptors like 45%, enzyme based, it is enzyme linked or enzyme coupled reaction, that is 28%, hormones and factors like 11%, ion channels like 5%, nuclear receptors like 2%. So if you see the receptors here, the component of a cell or organism that interacts with a drug, and it initiates the chain of biochemical events, as I said, cascade of reaction leading to the drug's observed effect. Then they have a specific binding site that are defined in size as well as in shape, and mostly they are present on or near the membrane. That is the very important part, on the membrane or near the membrane. So what happens on the membrane means if the plasma membrane is there, there itself it is there, or cell membrane is there, there itself the receptor is there. If it is not near to the cell area, it can be little bit down. So what happens, the upper part which is there, that can start to initiate the cascade of reaction, and this particular receptor can get activated. So some line in the enzymes or genes, that is a very important thing. When we will see about it, we will understand how the enzyme plays a very vital role here. And protein, that is a polypeptide in nature. So the receptors are basically the polypeptide nature. So protein receptors they are. So that plays a very vital role here. So if you see the history of receptors, it all started by Langley and Ehrlich, and introduced the concept of receptor. Langley, if you see in 1852 to 1925, what he studied was, studied the effect of atropine against pilocarpine induced salivation in cats. So that was the experiment what he did, and he postulated that there was a receptive substance in the nerve ending or gland cell with which both atropine and pilocarpine are capable of forming the compound. So that was his postulation or hypothesis, you can say, that he considered that there is a receptive substance which is present, and due to which they both are getting attracted or binded up at some place. Then Ehrlich, if you see, from 1854 to 1915, okay, so what he observed was that certain dyestuffs acted selectively, staining some cells more deeply or in a different way from other cells. So that suggested that drugs with selective actions on particular cells could be developed, introduced the term receptor. So he was a person who introduced a term called as receptor, and then later on everyone started using this particular term and it became very common in the pharmacology. So the four major families of receptors, if you see, as we have seen the percentages in the previous slide for the target. So the four main families of receptors are there. The first is called as these all are actually protein based receptors. So these are, the first one is ligand gated ion channels, like nicotinic ion channels. So the G protein coupled receptors, that is the alpha adrenoceptor, then enzyme linked receptors like insulin receptors, then nuclear receptors like glycocorticoid receptors. So the receptor that do not fall into these four receptor families, they have been categorized under some other different families, and they have been marked up like specific membrane ion pumps like sodium potassium pump, or you can say ATPases for ETC cycle ATPases, then specific enzymes like a 5-phosphodiesterase, then structural proteins like a collagen to tubulin, then cytosolic proteins, or that is cyclosporin to immunophilins, okay, immunophilins. So all these things are apart from the four families, they are also there for our movement. So if you see the four main types of receptors and how they are working, so type 1, type 2, type 3, type 4, we can categorize like this. So type 1 is the ligand gated ion channels, location is membrane, effector is channel mechanism is there. So coupling is by direct mechanism, and examples like nicotinic receptor, GABA R receptor. Then time taken for the cellular effect, if you see, it is milliseconds. Very easily it can go inside, okay, milliseconds. If you see G coupled receptor, that is membrane only, but effector is channel or based on the enzyme, because either it can be channelized or the enzyme will activate the cascade of reaction, then again getting back to the normal state. So this is there. And the G protein is the main important thing which is acting here as a receptor here. So coupling factor. So muscarinic receptor, adrenoreceptors, all these things, and it's like seconds only. But if you see enzyme linked receptors, that will take more time because that will be there inside. So it has to penetrate inside, then go inside, then act on the enzyme, and then start the cascade of reaction. So it will take some time. So enzyme linked receptors, like you can say, like it is an enzyme based. The effector is enzyme. Coupling is direct. Example is based on the insulin receptor, growth factors, and cytokine R, okay, hours. So that is why if you see the diabetic patient, they will have prolonged release return on their tablets. Prolonged release means it will take some time, like hours to like 1 hour to 2 hours. After that, slowly only it will be released in the channel, actually to say. Then nuclear receptors, it has to reach the nucleus, means it will take some time. So it is intracellular, okay, intracellular. So based on the gene transcription, it plays a very vital role in the gene transcription. And coupling is via DNA. And example wise, if you see, that is the steroid thyroid receptors. Then time scale for cellular effects, that is the hours and day here. So the first one in this category, it is called as type 1, that is a ligand gated ion channel. So it is coupled directly to the membrane ion channels. So agonist binding, that is opens the channel, causes depolarization, hyperpolarization, or changes in cytosolic ionic composition. Then depending on the ion that flows through. So it can be like from inside to outside or outside to inside. Both the things can happen in this case. So the control the fastest synaptic events in the nervous system. Synaptic events, you must be knowing that the shock like system. So that is actually taking place. So excitatory neurotransmitters such as ACT or glutamate cause an increase in the sodium potassium permeability. So ion channels, as I said, ion channels are working. So ion means sodium potassium ion channels. So they will start to open up. Sodium comes inside, potassium goes outside. So like that it can happen, and results in a net inward current. Net inward current means from outside it is actually going inside. So that will increase now and depolarizes the cell. So generate an action, that is the action which is potential. So receptors like nicotinic receptors, GABA receptors, glycine inhibitory, that is inhibitory AA, inhibitory of amino acids, excitatory amino acids like glutamate, kainate or NMDA, and 5-HT3 receptors are also present, which acts like in the same manner, ion gated channel. So what happens is in this particular channel also, it will just open up and take it up to particular movement. So you can see here how the process is taking place. Here, based on a signal molecule, that is the ligand. Ligand is acting like our signal molecule. So that will come and bind on our receptor. Once it comes and binds on the receptor, what will happen is the signal will be passed on to the plasma membrane and to the receptor that the channel has to be opened. So the ion channel gets opened because outside there are so much of ion is present outside. That is the potassium ion or sodium ion is more inside. So outside. So what happened? Once the signal binds on this receptor, there is a conformational change which occurs here. So what happens? It opens up and thereby it allows the ion. Once the ion is being reached, the required amount of ion is reached inside, then what happens is the signal will move out, and then what happens? Again, the conformational change will occur, which makes the receptor to come to the normal state and then closes up. So this is how the ion channel receptor is working here. The second receptor which is present here is called as G protein coupled receptors, or GPCR. So here it is having 7 alpha helical membrane spanning hydrophobic amino acid segments. So run into 3 extracellular and 3 intracellular loops. That is a very important thing. The outside you can see 3, inside you can see 3. So 3 loops are there outside. So that is how it is working. The binding of the mediator molecule induces a change in the conformational chain. So what happens? Once it comes and bind, it acts like a mediator. So what mediator is? Like a, you can say like a GDP. Okay. So GDP bounded one protein is there. So what happens? Once it starts to bind, when the signal gets, what happens? The GDP becomes GDP, it becomes activated, and it goes and activates the nearby enzymes. That enzyme will activate the cascade of reaction. Once the cascade of reaction is done, again it will go into the GDP state, from GDP to GDP state, and then what happens is there is a conformational change on the membrane also. So like this, the enabling to interact with the G protein lied at the inner leaf of the plasma lamella. Okay, so how it works? So G protein, if you see, the G protein consists of 3 subunits. That is alpha subunit, beta subunit, and gamma subunits. And in the inactive state, the GDP is bound to alpha subunit. So the activation lead to the displacement of the GDP by GDP. So what happens is, now the activated G alpha, which is there, the G alpha GDP dissociate from beta and gamma subunits, then associates with the effector protein and alter its functional state. So alter its functional state means from the ground state, it is going to the excited state now. So once it is going to the excited state, then what happens is the alpha subunit slowly hydrolyzes the bound GDP to become as GDP, because you know that when it is moving, energy is used. So once the energy is completely used, it will again go back from GDP to GDP state. Once it goes back to the G alpha GDP state, it again joins back with the beta and gamma subunit. And then what happens is the beta gamma dimer, which is there, can activate the receptor, opens the potassium channel, inhibit the voltage gated calcium 2+ channel, or promotes the GPCR desensitization at the higher rates of activation. So these all things can happen. So what is the different effects are there? The important G protein with their action on the effector cells or proteins, if you see, that is the GS state, that is GS is adenyl cyclase activation, Ca2+ channel opening, then GI, that is the adenyl cyclase inhibition, then K+ channel is opening, GO state, that is the calcium 2+ channel is inhibited or inhibition, GQ, that is the phospholipase C activation is taking place. So one receptor can utilize more than one G protein. Agonist pleiotropy. So that is example is you can see here. So muscarinic M2, they can work with a coupler of GI and GO. Muscarinic M1 and M3 with the GQ only. Dopamine D2, that is the GI and G0 or GO. Beta adrenergic, that is GS. Alpha 1 adrenergic, that is GQ. And alpha 2 adrenergic, that is GI and G0. So the 3 major pathways, effector pathways of the GPCRs, if you see here. So the first one is called as adenyl cyclase C-CAM pathway. So in this cyclic AMP pathway, so in this what happens is the activation of the adenyl cyclase result in the intracellular accumulation of the second messenger, that is the cyclic AMP. Now, since the concentration of the secondary messenger, that is the CAMP level, is higher, so what happens is the CAMP functions mainly through the CAMP dependent protein kinase. So when the kinase is coming, the phosphorylation is taking place. So the PK phosphorylates, and thereby what happens? It alters the function of many enzymes, ion channels, transporters, transcription factors, and structural proteins. Second step in this is phospholipase C IP3 DAG pathway. So in this what happens is the activation of the phospholipase C hydrolyzes the membrane PIP2 to generate the secondary messengers IP3 and DAG. So due to this what happens, the inositol, that is the IP3 inositol triphosphate, that is the IP3, it is diffusing to the cytosol. And due to the diffusing to the cytosol, what happens? The CA2, which is there, that also mobilizes from the endoplasmic reticulum deports. So it starts to mobilize. From there, it starts to move out. Okay. Then diacyl glycerol, DAG, what it does? Remains within the membrane, recruits a protein kinase C, that is the PKCs. So what happens here? So activates it with the help of the CA2+. So what happens? It channelized to go up a little bit. So activated PKC, what they do? Phosphorylates many intracellular proteins, mediates various physiological responses. So what happens now? Triggered by the IP3, the CA2+ is released, almost release the CA2+, mediates and modulates. So what are the things which is done? Contraction, secretion or transmitter release, then eicosanoid synthesis, then neural excitability, then membrane function, metabolism, all these things occurs because of the particular function here. So the channel regulation, how the channel regulation is taking place. As we have seen in the previous slide, there are almost like different things like GI is there, GS is there, GO is there, how it works. Okay, so the activated G protein, how they work is can open or inhibit the ionic channel specific for the CA2+ and K+. So what happens here? Since the channel is getting inhibited, once the channel is inhibited, what will happen? The function will not take place. So either they can open up the channel or they can stop the channel or inhibit the channel. So without the interventions of any other second messenger, so if there is no second messenger is there in between the coupled reaction, there is no second messenger is there like IP3, inositol triphosphate or CAMP, nothing is there. Then what will happen? The process will stop there itself. In the first step itself, it will stop. So there is no secondary messenger due to that reason. So the hyperpolarization, depolarization or changes in the intracellular CA2+ can occur when the secondary messenger intervention is taking place. So the GS is there, what it does? Opens the channel in myocardium and skeletal muscles. So it is helping in the myocardium and skeletal muscles. GIGO. So GI is for inhibition, GO is for opening. So GO, that is opens the K+ channels in the heart and the smooth muscle. GI, if you see, that is the inhibiting the neural CA2+ channels. So this function takes place. Then direct channel regulation is mostly the function of the beta and gamma dimer formation. So these are the different major functional pathways of the G protein receptor transduction. So you can see the examples here in detail that all the pathways, whatever the different pathways are being given, or CA2+, how it goes up or comes inside, or potassium, how it is going out. So all these things can be shown here. So this is how the GPCR looks like. As we have said, 2 loops are there outside and 2 loops are there inside. So extracellular 2 are there, intracellular 2 are there. So these are actually the G protein binding site. G protein binding site means you are having your alpha, beta and gamma in this position only. So how it works? So you can see here, consider this is your G protein linked receptor. The previous slide, what we have seen, this is here. Okay. Now this is our receptor which is present here. So now what happens? The chemical molecule which is there, that is acting like our signal, that will come and bind here. Once it comes and binds here, so what happens here is it interacts with it, and due to the interaction, the GDP which is present here, that is getting phosphorylated. So once it gets phosphorylated, it becomes from the G protein, that becomes as GDP to GTP. So the alpha protein, the G alpha protein is actually now GTP. So once it is GTP, the membrane bound enzyme which is there, that will be attracted by this GTP. So what happens? That will move towards the GTP, and the GTP with the G alpha will move towards the enzyme. There will be a lock and key mechanism by which the enzyme will get activated. Once the enzyme is getting activated, the series of reaction will start inside, and once the series of reaction is done, it will again undergo a dephosphorylation, by which what happens is the phosphate which is there from the GTP, it again becomes as GDP and again gets released, and thereby the signal molecule also get released from the particular site. So third type of receptor, if you see here, this is called as enzyme linked receptor. So enzyme linked receptor, if you see, this is like lie partially outside, or you can say partially inside the cellular membrane. So consisting of the extracellular ligand binding domain linked to intracellular domain by single transmembrane helix. So single transmembrane helix means from the protein there is a helical structure which is binding this protein enzyme and keeping it. So the intracellular portion is enzyme in nature. So intracellular is enzymatic in nature. So the protein kinase generally they will work. So phosphorylation will easily take place here. Guanylyl cyclase in some cases also help for this process. So what is happening here? The commonest protein kinases which is present here is receptor tyrosine kinase, that is called as RTK. So what happens, as I said, they phosphorylate. Wherever the kinase comes, it is going to phosphorylate. So RTK is going to phosphorylate tyrosine residue on the substrate proteins. So due to this, what is happening? The different receptors are there, like insulin, epidermal growth factor like EGF, nerve growth factor like NGF, and many other growth factors receptors are present here. So how they are going to work? So consider this is our tyrosine kinase which is present here. Tyrosine is there. So now what is happening is the ligand which is there, that is open. Okay, so the site is there open. So now what is there? It is going to bind. So once the signal molecule is binding, the ligand is going to bind on the receptors. They form a dimer. They form a dimer means they will come closer. Okay, so they form a dimer. Once the dimer is formed, then what happens? These two molecules which is there, that is the tyrosine kinase receptor, they will come closer. So there is a conformational change by which the dimer has formed. So consider this is the dimer which has been formed now. Once the dimer is formed, now what happens? This grooves, what you see here, that is getting activated. So by the phosphorylation, the ATP which is there, the tyrosine on the tail of the other polypeptide, they will attract it. Okay, so the enzymes which are there, they will come and bind to this, and then what they do is they will start the series of reaction. Once the series of reaction is done, the signal molecule which is there, that will get released. So once the signal molecules are released, they will move out. Okay, so everything will move out, and then what happens is this will be separated. So this is how the enzyme linked kinase receptors are working. The fourth receptor in this category is called as nuclear receptor. So nuclear receptor means, from the name itself we can know it is intracellular. It is not on the membrane, it is intracellular. So intracellular, that is a cytoplasmic or nuclear soluble proteins which respond to lipid soluble chemical messenger that penetrates the cell. So here what happens is, when the hormone binds to the receptor protein, the receptor dimerizes, the same manner dimerizes, and the DNA binding regulatory segments folds into the requisite configuration. So due to this dimer formation, what is happening is moves to the nucleus. So this particular thing, they will move into the nuclear pore and they go to the nucleus. So binds with a co-activator or co-repressor protein, which have a modulatory influence on its capacity to alter the gene function. So what happens? The gene function which is there, that will be activated or it will be inhibited. So here, the main inhibition will not be replication. The main inhibition will be either the transcription process or the early, you can say like the splicing process, or the translation will be blocked. So that is how the process is being inhibited. So that is, we have to understand here. So the whole complex, what you see here, that is attaches to the specific DNA sequence of the target genes, facilitate or repress their expression. Specific mRNA is synthesized or repressed on the template of the gene itself. So the transcription is blocked here or it is activated. So the mRNA directs synthesis of specific proteins which regulates the activity of the target cells. Example for this is corticosteroid, sex hormones like testosterone, progesterones, estrogens, all these things, and thyroid hormones. So you must be knowing about the hormones like testosterone. Many people for bodybuilding, they use testosterone. What happens? This hormone, this will be activated inside the cell. That is why the persons who take this, sometimes if they stop it, their testosterone levels will be at a high peak. If they don't do exercise and all, that will be actually giving a side effect. That is, they will be either very hefty, it is going to affect some other things, or metabolism also sometimes are affected. So some side effects can occur because it is taking place intracellular. Okay, so stimulates the transcription of the genes by binding to the specific DNA consequences. So this is the process by which it's taking place. So in the cell, inside the cell, you can say like epigenetic level. So the gene is there. So since you can see here, this is our ligand, which is coming inside. It is binding to the particular receptor. Once the receptor is formed, they formed a zinc finger-like structure. So they formed a dimer. So due to the dimer formation, they are actually binding and they are fixing to the particular region. So due to this, what is happening? The transcription is taking place, or it can inhibit the transcription based on this particular dimer system itself. So due to this, what happens? A particular molecule or enzyme will not be synthesized, or particular enzyme or molecule will be synthesized. So both the things can occur, and that is going to help us. So these are the different references from which we have taken this particular notes. So if you have having any doubts, you can take up these books and you can refer it, and you can read these books for the particular topic. I hope that you have got an idea. So thank you and have a good day.